Hello everyone, my name is Elizabeth Evans and today I'd like to share with you part of our research using micro-CT imaging techniques for visualising pinniped mystacial pad musculature. This work is led by Dr. Alex Elder of Manchester Metropolitan University in collaboration with the NXCT at the University of Manchester. Pinnipeds are a group of mammals which include seals, sea lions and walruses. And here is an example of a harbour seal, which is the species we used for the data I am presenting today. All pinniped species rely on their whiskers for foraging, hunting and navigation, and these whiskers form part of a well-developed sensory system that vary across the different pinniped species. However, description of the muscle structures which control the whiskers is scarce within the literature. This is because the traditional method of analysis for other whiskered mammals like rodents uses histology, which has its limits, particularly with regards to specimen size and the number of slices which can be made. Here's an example of the whisker muscular structure from a rat, visualized using histology. These muscles here, around the follicles, control the whisker movement and are similar to the muscles of interest that we wish to find in the seals. But rats and small rodents also have less curvature to the muscle pad, meaning it can be easily sectioned in a way that captures all the whisker follicles and the associated muscles, just like here on the screen. Pinnipeds are much larger mammals with more curved mystacial pads. Another drawback of histology is, of course, that it is destructive, meaning that studying museum specimens with this method is not possible if the samples must be returned to the collection undamaged. We therefore chose to use X-ray microcomputer tomography as the method of analysis because it is a non-destructive method of acquiring 3D morphological data. So, the aims of this study were to visualise the mystacial pad muscles from harbour seal specimens, optimise the scanning method for future samples and establish a repeatable and adaptable data analysis workflow. So this is what a typical sample setup looked like during our study to ensure the samples didn't move or dry out during scanning. The samples came from the National Museum Scotland, and because they would be returned to the museum collection, we wanted to see if we could scan the samples without additional modification like staining. So initially we scanned the samples simply fixed with formalin at a suitable scan setting for the tissue type. Unfortunately, as you can see, the contrast between the major features we wanted to visualise is very poor, even after trying several different scan settings, and this dataset is just one from the unstained datasets. To show this issue in a more quantitative way, using a line profile, it's very difficult to separate the edges of the features within the image data. We therefore decided to employ a reversible tissue staining technique called dye-CT, which was developed and popularised by Paul Gignac. Here's an example from Dr. Gignac's paper, which is a dye-CT stained western diamondback rattlesnake head. This technique enhances contrast between soft tissue structures using Lugol solution, which is a solution of iodine and iodate salts. Because the iodine changes the radiodensity of the tissues, we had to try other scan settings that were a much higher voltage than the unstained pads. The final settings we chose compromised on pixel size, as we could have achieved more magnification, but these data provided good contrast and low noise. However, this may mean we missed some very fine structure that was present. With our final CT settings and sample preparation method in hand, we could now use these as a starting point for future work on other pinniped species, bearing in mind that some variation to the settings will be needed to account for sample size or ratio of tissues more likely to readily absorb the contrast agent, including the external fur, which is something you can see here on the stained image. The next challenge was to analyse the data. The most straightforward image data of any type to analyse is that with high contrast between the major features with minimum overlap in the grayscale values. Unfortunately, biological samples, like our example here, has a large amount of overlap between the grayscale values of the features of interest. However, we can see the shape and texture contrast easily with the human eye. So to improve the shape and texture contrast, we can see by eye, but so we can digitally segment the data. We want to still reduce the noise further and smooth the grayscale values while preserving the edges of the shapes. As a result, we tested a number of image filters on the data, and the final filter we chose was a non-local means filter, which preserved the edges of features while smoothing noise. Using our knowledge of the muscle structure from other mammalian species like rodents, we could select regions from the image and build up the 3D morphology of the muscles and the whisker structures. And we did all this using a combination of automated and manual approaches in Thermo Fisher's Aviso and Mirror software. Here is an example of a combined structure in 3D. So we have in white the whiskers, with the follicles in yellows separated by collagen sheets that create rows in the mystacial pads, and then in pink is one of the major muscle groups involved in the overall movement of the cheek itself. And you can see here the one-to-one of the yellow of the follicles can be seen here in the 2D cross-section, and the same, the white of the whiskers is the bright white centres of the follicles, and these sheet structures here in 2D are seen here in red in 3D. Although we successfully imaged the target muscle structure, it's worth highlighting some of the limitations of the study directly related to our methodology. Firstly, CT is a costly analysis technique with scan times on the order of hours to achieve the data quality we wanted. As a result, this limits the number of samples which can be analysed, and this naturally raises questions of repeatability of the work and the ubiquity of the structures we have seen in a single individual. Our data processing methods and chosen resolution managed to identify what we believe to be the larger scale muscle structures which control the whisker movement. However, as I touched on earlier, we may have failed to resolve finer features in the data. This is a limitation of any imaging technique, but it bears mentioning as we can only measure what we can detect. A third point to highlight is that although the DICE-CT method is provably extremely powerful, it can introduce artifacts from both understaining the sample and if the samples become overstained. Overstaining artifacts are a particular problem as they can cause tissue shrinkage, which modifies the size and the shape of features. All that said, DICE-CT offers superior soft tissue imaging quality compared to traditional CT methods. So, to close, to understand the sensory anatomy in pinnipeds, we need to be able to accurately identify the muscle structures in the mystacial pads. Micro-CT has proven to be an invaluable tool for understanding the anatomy of biological samples, both in vivo and preserved specimens like ours. Having our samples preserved allows us to use the reversible staining technique, DICE-CT. As we have shown, the DICE-CT significantly improves the data we can obtain compared with standard CT scanning, leading to our group being able to identify the key muscle structures of interest in 3D. Many thanks for listening, and thanks again to Dr. Alex Elder and to our collaborators at the National Museum Scotland, Manchester Metropolitan University and the University of Manchester.